Our today's lecture is based on centrioles, which are included in cytoskeletal structures of the cell. Centrioles were discovered by Boveri in 1888, and centrioles are present only in animal cells. In case of plant cells, fungal cells, gymnosperms, angiosperms, these are absent. You will not see centrioles in plant cells. These are only present in animal cells, where they are essential for cell division. Usually, if we take a cell, let this be a animal cell. This is nucleus. And this is cytoplasm. In the cytoplasm, we will see two centrioles. A pair of centrioles is there, just like this, at right angles to each other. A pair of centrioles is called diplosome. Diplosome, a pair of centrioles. That means each cell must have two centrioles, a pair of centrioles, but when there is no function, they mainly help in cell division. These two centrioles are a diplosome. These are enclosed by a layer of cytoplasm, which is called centrosphere. Centrosphere. These are pair of centrioles called diplosome, and this is centrosphere, which encloses them, which covers them. It is actually a piece of cytoplasm which covers them. And now this structure has centrosphere and two centrioles or diplosome, and usually it is called centrosome. Centrosome. Centrosome includes diplosome plus centrosphere. Once again, I will repeat. Each cell has two centrioles. The centrioles occur at right angles when there is no function. A pair of centrioles is called centrosome. A pair of centrioles is called diplosome. The diplosome is covered by a piece of cytoplasm which is called centrosphere. The cover is called centrosphere. And whole structure, centrosphere, two centrioles or diplosome, is called centrosome. So we usually say that cells contain centrosomes. Actually, these are centrosome. Actually, these are centrioles. And we will take individual this centriole now, and we will study its structure. Individual centriole. Centriole, if we will take, we will have to study its transverse section. The diameter of each centriole is 0.1 to 0.2 micrometers. This is diameter. 0.1 to 0.2 micrometers, and we will see their transverse section. The transverse section shows a cartwheel structure. It is called a cartwheel structure. Cartwheel, you know, resembles a cartwheel structure, and the basis of centrioles is microtubules. Microtubules, I have explained you in my previous lecture what are microtubules, how are these formed. Microtubules are formed of 13 protofilaments. I have told you that time that each microtubule has 13 protofilaments, and each protofilament is made up of a zigzag structure of alpha tubulin and beta tubulin or tubulin dipolars. Now, if we will take the transverse section of a centriole, a cartwheel structure, just as I will draw here. This is periphery, center is black. This is black. This is their periphery. In the periphery, there are microtubules now. Now microtubules are in the form of triplets, just like this. This is microtubule E, which is complete. That means complete means it has 13 protofilaments. It is known as complete. Then B. B is incomplete. That means it is one part has no microtubules. If this has 13 protofilaments, this has only 10. And C. C also has 10. These are also called subtubules A, B, and C. A is complete. It is these are 3 microtubules. These are called microtubule triplets. Triplets. Triplets means 3 microtubules are associated with each other. A, B, and C. A has 13 protofilaments. B has 10 and C has 10. So A is complete, B and C are incomplete with only 10 protofilaments. And this forms the association of triplets A, B, and C. We have to arrange 9 such triplets in the periphery. This is periphery. 9 such triplets A, B, and C, just as this. If you draw, this is A, slight inclination. This is B and this is C, slightly inclined. A, B, and C. Similarly here, A, B, and C. A, B, and C. Similarly, A, B, and C is similarly here. A, B, and C. These are 1, 2, 3, 4. This is 5th triplet. This is 6th triplet. This is 7th triplet. This is 8th triplet. And this is 9th triplet. So 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. A, B, C, A, B, C, A, B, C, A, B, C, A, B, C. Now, A is connected with C. This is called AC linker. AC linker. Linker means it connects. It is a protein which connects. Here A is connected with C. 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 Connected with C. A is connected with C, and A is connected with C. So A is connected with C. This is called, each A is connected with C. A is slightly on outside, C is slightly on outside. A is slightly on inside and C is outside on outside. Now the center. This is center. Center has a cytoplasmic circle, but there is no centriole in it. There is no microtubule in it. It is called hub. Center is called hub. It is black. Black means there is no microtubule here. So 3 microtubules in 9 microtubule triplets are here. So we say that the arrangement of microtubules in a centriole is 9 plus 0. You should note it. 9 plus 0. That means 9 triplets on the periphery and no microtubule in the center. Each A is connected with C by AC linkers. Now, A is also connected with hub. This is hub. Just like this. A is connected with hub. This is called radial support. A radial support, just as you have seen in motorcycle or cycle. Radial support or radial fiber. radial suppork or radial fiber. And near A, this radial suppork has a small protein which is called suppork head. This is suppork head. It is per strand. Suppork. And you should know here that there is no membrane. It is not covered by a membrane. This forms the cartwheel structure of centriole. So, each centriole is formed of microtubules. The arrangement of microtubules is 9 plus 0. The microtubules are in the form of triplets, also called subtubules, named as A, B, and C. Now, how will we recognize them? A is complete with 13 protofilaments, B and C are incomplete with 10 protofilaments each. These microtubules are arranged in the periphery of centriole. Now A is connected with C by AC linker. The center has no microtubule, and I have told you the arrangement is 9 plus 0. The center is called hub. It has a protoplasmic sheath in the center. The hub or protoplasmic sheath is connected with a microtubule each, which is called radial suppork or radial fiber, and which has a radial suppork head. This is suppork head. It is a protein actually known as suppork head. The suppork head is present near the E. This is the structure of centriole. The main function of centriole is cell division. And when we will talk about mitosis, cell division. For cell division, it is important in animal cells. It helps there in cell division. So this was the structure of centrioles and their function. Thanks for today.